on metagenomics. This introductory webinar will highlight applications of metagenomic sequencing and methods of analysis for metagenomic data that are used in various research, biomedical, and agricultural applications. Before we start speaking about metagenomics, I wanted to introduce you to Omics Logic programs in Africa, and more specifically, a new partnership with Helix Biogen Institute that will help more students and researchers gain access to Omics Logic training and research solutions in Nigeria. To speak about the significance of data literacy and bioinformatics in Nigeria, I would like to invite Dr. Oladipo Elijah Kolabole, the founder of Helix Biogen Institute and a lecturer at Adeleke University. Dr. Kolabole has a PhD in microbiology and a master's degree in medical microbiology and parasitology. In the past, he has been a lecturer and research director of molecular biology, immunology, and bioinformatics research laboratory, and now continues to participate in bioinformatics training across Nigeria and around the world. Dr. Kolabole, over to you.
Yeah, good afternoon, everyone. It's a nice privilege given to us to be here and to collaborate with our partners, Pine Biotech, T-Plat Platform, and Toba Bioinformatics. Helix Biogen Institute is an upcoming institute in Nigeria that is involved in capacity development in the area of bioinformatics. And we have been running a lot of courses like a certificate diploma. And now we are having a collaboration with our partners now in order to allow Nigerian scientists, both young and old, African scientists, both young and old, to know much about bioinformatics and how to use various tools in order to solve real life problems. And I want to believe that as we are commencing this program, we will know each other better. And I want you to know that the facilitators for this program, they are here to give you the best, give you what it takes to know much about bioinformatics. And as I believe that at the end of this seminar, we all will be interested to move further to the next stage into the retraining. And thanks everyone, we are welcome.
Thank you so much, Dr. Kolabole, for organizing this workshop. I would now like to say a few words about the Omics Logic team and our collaborators who have made this training possible. My name is Beepsha. My name is Beepsha Biswas, and I am the Omics Logic Community Manager responsible for daily interaction with thousands of users that are part of our bioinformatics community managed by Pine Biotech. Over the course of the past several years, our team has been collaborating with a number of academic institutions around the world that have participated in content evaluation, curriculum review, and project design. As a result, the Omics Logic programs have been thoroughly evaluated and are known to lead to excellent outcomes. The goal of the Omics Logic programs is to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. Omics Logic Metagenomics training program will be guided by experienced trainers that have ample experience with bioinformatics in both academic and industry applications. I will now invite Ilya Brodsky, who is the CEO of Pine Biotech, to speak about metagenomic sequencing and the upcoming program. Over to you, Ilya.
Thank you, Beepsa, and thank you everyone for joining today's webinar. So what we are going to cover in this webinar is a little bit of an introduction to metagenomics, sequencing, and how this data could be analyzed and used so that in the context of the upcoming program, you will have a sense of what we will do together. So let's start from the very beginning. The connection between disease and microorganisms has been established in the 1800s, and already towards the end of the 19th century, laboratory methods for growing microorganisms in a lab culture were developed, helping characterize them with staining. These studies provided important information about bacterial function in isolation, but it was soon apparent that most microorganisms actually live in complex communities. And today we know that the microorganisms or the microbiota, communities of microorganisms that make up the human microbiome, are not just unicellular organisms living alongside each other, but instead they form highly regulated, structurally and functionally organized communities that are attached to surfaces as biofilms with interspecies collaboration and antagonisms that contribute to ecologic stability. Now, many of us are probably familiar with this stability. It's fascinating that we know, for example, trying to battle with such common skin conditions as eczema, acne, and dry skin. So these communities are distinct and connected, and they are determined to maintain their community the way that it is. And bacteria within a biofilm can communicate with each other by producing, detecting, and responding to small diffusible signal molecules in a process called quorum sensing, which confirms benefit for host colonization, biofilm formation, defense against competitors, and adaptation to changes in the environment. Quorum sensing activities in biofilms are also involved in the virulence and pathogenic potential of bacteria and are therefore an important factor in understanding and controlling bacterial infections, as they enable microorganisms and biofilms to become more tolerant of host defenses and antimicrobial agents. So microorganisms are living in communities, forming specific structures, and those structures help them maintain that community by leveraging different characteristics of the bacterial species found in a given microbiota or community of bacteria. The human microbiota is estimated to be 10 to the 13th to 10 to the 14th of microbial cells. So it was previously thought that there are many more microorganisms in the human body compared to the human cells, but today we know that approximately it's 1-to-1 ratio of microbial cells to human cells. And these numbers are primarily derived from the total bacterial cells in the colon, which is the organ that harbors the densest number of microbes. However, each one of these microorganisms carries important functional properties and genes that contain genomic information. These properties and the genomic information about microorganisms have been of a growing interest to the scientific community. And so now there are over 80,000 publications on the microbiome, and this number is continuously growing, showing the significance of microbiome in a number of different applications. However, the majority of biomedical studies on the microbiome have up until now been about the gut microbiota, but we'll see how major projects and ongoing studies are expanding the role of microbiome in a variety of different applications. So since the 1990s, microbiome studies have significantly advanced our understanding of different types of microbiota. For example, we see here metagenomics of the human intestinal tract and the subsequent human microbiome project. These are huge studies that have been made possible through the advances in metagenomic sequencing. As a result, we now know that there is a distinct skin, lung, gut, and even brain microbiota that are characterized in the context of healthy, but also diseased conditions. And so over the past decade, more than 1.7 billion US has been invested in human microbiome research. Major projects are underway in the United States, the European Union, China, Canada, Ireland, South Korea, and Japan. And this investment has really showed and confirmed the importance of the microbiome to human health and human development. Some 20% of US investment in this research has gone to 2 phases of a large project called the Human Microbiome Project, or HMP, which is creating the research resources needed to study the human microbiome, both in healthy development and in diseases. And one of the major goals of HMP, which was launched in 2007, was to create a toolbox of reference data sets, computational techniques, analytical methods, and clinical protocols. So let's talk a little bit about what is known about the microbiome function for humans, for people. So today we know that endogenous human microbial communities contribute to a critical metabolic, physiological, and immunological function, including differentiation and maturation of the host mucosa and its immune system, food digestion and nutrition, energy generation, metabolic regulation and control of fat storage, processing and detoxification of environmental chemicals, maintenance of skin and mucosa barrier function, development and regulation of the immune system, and prevention of invasion and growth of disease-promoting microorganisms. So to study these functions and how they vary in different conditions, there have been a number of technologies that have been instrumental to our understanding of how these communities live, function, and develop and respond to different stresses. So these questions are primarily being answered by 2 types of technologies. One is called sequencing, so whole genome sequencing or whole DNA sequencing, or metagenomic sequencing or metatranscriptomic sequencing, as well as different types of amplicon sequencing, which we'll talk about in just a few more slides, and mass spectrometry. So mass spectrometry allows us to detect spectra of proteins and metabolites and really understand what type of functional enzymes and molecules are being produced by these microbiota. So here we'll talk about how these techniques are used and try to understand one of the major functions of metagenomic sequencing, which really allows us to understand the communities as a functional group. And so we'll talk a little bit about how the microbiota could be sequenced, mapped onto a database of reference genomes of all known bacterial genomes, and give us a sense of species richness. So in this context, metagenomics is really different from other types of omics data. So typically when you think about omics data, you're thinking about genomics or transcriptomics, and all of these focus on the whole of a single organism. And organisms, like metagenomics is the community of microorganisms, they live in communities. And so when we think of metagenomic sequencing, we're really trying to understand the composition and the structure of that community. And so this genetic material is sampled directly from an environment, so it skips that stage of isolating a particular strain and allows us to understand how do they interact with each other and their natural environment. And so when we think of metagenomics, we can really think of them in 2 ways. So the first one could be applied to environmental culture-free sequencing to study populations. And we can also use sequencing to extract a particular strain and understand how it relates to other types of known genomes that we have characterized. So these 2 types are really sometimes used in a complementary way. For example, if we want to look at the gut microbiome, we want to ask the question, what types of bacteria and in what proportions are they inside this particular individual? And we might want to compare that individual to another individual. And sometimes we want to understand how a particular member of this community, behaves and what's different about it. For example, some kind of a antibiotic resistant bacteria or microorganism, and then we can use full genome assembly and mapping to understand how it could be mapped to known species. So the first type, which is really 16S ribosomal RNA metagenomic sequencing, is a low-cost and flexible approach to study how the composition of a particular microbiota could be characterized. So this kind of a sequencing allows us to essentially extract a 1,500 long base pair sequence of the 16S ribosomal RNA gene, map it onto a database where we have organized all of our data into operational taxonomic units through pairwise, pair-ended sequencing. And by mapping, we are really understanding how a taxonomy of a particular microbiota explains a particular microbiota, a particular defined community. So what is the 16S ribosomal RNA gene? You have the bottom ribosomal subunit, or the 30S small subunit of the ribosome that is specific to bacteria. Inside that, you have the 16S ribosomal RNA gene. So this is actually RNA. And that could be sequenced using a specific primer so that you're only sequencing for particular hypervariable regions. Now, a lot of times, hypervariable regions 3 and 4 are used, and by mapping those onto a known sequence of 16S ribosomal RNA, we have a particular sensitivity to different levels of taxonomic classification. So we can ask the question, for example, what you see here is based on phyla of all species that are present, what is the proportion of different phyla in different places of the body? For example, you can see here skin, oral cavity, etc. And now you can ask the question, well, how can we analyze this data? And what are some of the approaches to ask specific research questions of the data that we have generated using next generation sequencing? So an important part of this is to really understand the relationship between different microbiota and the relationship between the host and whatever that community contains, which could contain a number of members, for example, bacteria and viruses, right? So that those could be pathogens and they could be helpful bacteria. And so we want to, for example, know the composition, how many are viruses, how many are bacteria, or what types of viruses are present, or what types of bacteria are present. We might want to understand the relationship between the host and the microbiota. For example, changes in microbiota, how do they affect the host? Or if we have specific clinical or phenotypic data, for example, disease stage and not disease stage, susceptible and not susceptible, how do we characterize those types of interactions and changes across different conditions? So ultimately, the role of bioinformatics and different aspects of bioinformatics is to help establish those relationships and help us identify specific types of relationships that could be applied to the development of biomarkers and an understanding of specificity, features, and result of changes in those features as we think about diagnostic and therapeutic applications. So let's take a look at a few examples. And by the way, all of these examples are taken so that we can understand and anticipate what kind of training is going to happen when we turn to the Metagenomic, Omics Logic Metagenomic program. So what are project examples? When we say project examples, really what we mean is curated data sets, digested publications, and guided tutorials that we're going to include in the Omics Logic Metagenomics training program so that any one of you without or with some background in bioinformatics and in microbiology can understand how this metagenomic sequencing data could be analyzed for some kind of a valuable application. So what are those valuable applications? One example is a project called the American Gut. And even though it's called the American Gut, this is a part of the Human Microbiome Project. So in fact, international participants have contributed their samples to a group that studies those samples by sequencing them and asking survey data to establish some general trends about the way that the people that are reporting, for example, sensitivities or their level of activity or their background, ethnic background, and their location or maybe specific food diet, we can understand variation of healthy human microbiome. So one of the examples that we'll do is we'll take a number of representatives from the American Gut Microbiome Project, and we're going to prepare a report. So this is what you see right here. This is an actual report from the American Gut that shows how a particular user that has submitted their samples, what are the most abundant microbes, most enriched microbes, how does your microbiome compare to others, and maybe some specific examples of different groups, people that are more active and less active. And so we'll ask the question, what are some general trends for microbiome variation in healthy human population? Another important application of microbiome is something that is a little bit less intuitive. So we'll take a look at a project that tries to separate the influence of microbiome changes from the overall environmental conditions that have an association with different types of behavior. So we'll take a look at an animal model study where animals were fed with a particular diet, a high-fat diet versus a normal diet, and then that microbiome, once it has been established, was transplanted into another set of animals, and then their behavior was studied. And so through those kinds of studies, we'll see how you can observe a relationship between microbiome composition, presence of particular bacterial phyla or species, and the composition of it. And then you can identify potentially an interesting insight into how anxiety and behavior could be affected simply by microbiome. Another important application of microbiome that we'll look at is the way that the microbiome and the food that the microbiome thrives on determines efficacy of treatment and our response to different types of triggers, psychological triggers, behavioral triggers, etc. So we'll see at the way that these types of data sets could really be informative and lead to development of intervention. So we'll in fact look at one example where a treatment using microbiome from fecal samples was approved in China for Alzheimer's disease. So really interesting applications of microbiome in terms of treatment. But we'll also look at the impact of the microbiome on treatment response. So how microbiome composition of individual patients can have an effect on their toxicity and beneficial treatment metabolism. So we'll look at microbiome as an important layer in that process of taking a treatment, taking a drug, and essentially what happens, how effective is that treatment is also determined by the microbiome, which is logical, right? Because we just saw how the microbiome is related to the immune system and it forms a biofilm essentially on the layers of our body that are exposed to these treatments. But of course, microbiome is not only useful in biomedical applications. It also has a huge benefit for agriculture. And so we'll see how microbial communities from the soil, from the water, have a major impact on the growth and the productivity of particular types of crops. For example, we look at microbial fertilizers, which are an emerging class of organic fertilizers and consist of dried microbial biomass, for instance, produced on effluents from the food and beverage industry. So they have been shown to provide unique benefits, especially in harsh conditions, and at the same time with minimal environmental impacts. And metagenomic studies on agricultural applications of microbiome are really showing promise in terms of our understanding of the role of microbiome, but also our ability to develop novel applications that are designed to make the crops more productive and more resilient in harsher conditions that are coming as a result of climate change. So now to explain how all of these topics are going to be covered in our upcoming program, I will pass it on to Dr. Mohit Mazumdar, who is going to explain what the program consists of and how to register.
Thank you, Ilya, and hello, everyone. So let me share my screen. So Ilya, can you make me co-host? I have another login, Dr. Mohit Mazumdar. Ilya?
Yes, sorry.
Yeah, thanks. Okay, hello, everyone. And my name is Mohit Mazumdar, and I'm one of the global representatives of Pine Biotech. And in my current role, I'm working with scientists, researchers, students to assist with their big data research. So talking about today's session and this program itself. So Omics Logic Metagenomics is a specialized program that not only takes you through the live sessions, courses, and example projects, but also get you to learn by applying this knowledge to a real metagenomics project. So as in in all of our Omics Logic training programs, we will leverage online interactive sessions, self-guided study materials, and that are crafted, designed with practical examples. So we'll go through practical assignments for an immersive experience that has proven to be effective for thousands of students who have already completed our Omics Logic training programs. So this online program is basically designed to be equally rewarding for students and researchers, and especially suited for project-based learning on a flexible schedule. So this program will cover main aspects of metagenomic sequencing, analysis of 16S rRNA, and other aspects of microbiology research using bioinformatics. So 16S metagenomics data processing would result in a table of annotated 16S rRNA genes, and there will be read map counts. So the table includes annotation that would have like levels of kingdom, phylum, class, order, family, genus, and species, and things like that. So we will be covering those towards the beginning, where we will be doing the first session covering introduction to metagenomics on 16 February, and then mapping to different databases. So how we can do the annotation and find those using different databases that are available. Then, as Ilya discussed, that we will be going through the data to pipeline, and there we will be using other tools such as PhyloSeq, and then the projects. So after going through the couple of the introductory session and understanding how to apply metagenomic sequencing into research or understanding by project examples, we will be going through the gut microbiome project, human microbiome, to understand the human microbiome variability. And followed by that, we will have another project session on the gut brain axis. And then we will be learning about the independent project. So the process and towards the end of this program, participants will be conducting independent or group projects, analyzing data from public domain, and learning from high impact biomedical publications on microbiome. So in order to study microbiome or microbial communities or microbiomes, we will be learning about a couple of technologies like we discussed, like we will be learning about 16S and 18S ribosomal RNAs, and the way to analyze the hypervariable regions that can yield important information about the microbiome composition. So the sessions will include asynchronous materials which have all the information, like the things that we have discussed. They are well described in the courses. So good understanding of the way data is being prepared, we will understand through the courses and the asynchronous projects, and how this data is helping us and allowing us to navigate research studies with ease, that we will be learning through the asynchronous courses, and that will be showcasing you in a while, that these introductory and hands-on courses, along with the educational project that you will have access to throughout this time. So other than the online sessions throughout the week, we will be going through courses and online projects. So this will be done via the Zoom session. So as this session is ongoing right now, we will have Zoom sessions throughout this entire program starting from 16 February. And the sessions will be held at the same time, 5 p.m. to 6:30 p.m. Western Africa time. And we will have a team of supporters to be able to help you transition this knowledge from theoretical knowledge to practical assignments, to applying this into a research project. So this entire process will be guided by us, and also will be getting help from our Nigerian team, whom we will be introducing in a while. So during the last program, or also during this program, once we transition, so the first part would be to learn from the theoretical aspects and learn about the basics, and then go through the practical assignments and go through the data sets and learn how to apply them. And then during the end of the program, towards the end of the program, we would ask you to submit a research proposal. So that research proposal that would have a unique idea, or an idea that you wanted to work on, and that you have discussed with the mentor, and and that you are sure about the analysis process and identified the data set, and also the analysis methodology that you will be taking. So we will be peer reviewing that proposal further with our experts, whom we have collaborated in the university, and also Tauber Bioinformatics Research Center, to be able to get peer-reviewed feedback, so that we can improve our research and move in the right direction. So this whole week towards the end will include one-on-one meetings with the mentor to work on the proposal, and also we'll have group presentations where the participants will get a chance to talk about what they have learned and what they understand from their point of view about the research question and the analysis that they wanted to do. So we have been doing it for a while. So our Omics Logic Metagenomics program has been taken by hundreds of researchers. That includes researchers who starts from the very basic without any background of bioinformatics, and also researchers who come from a background who understands and knows how to apply these technology to a research question. So there are different expectations that we have seen from different categories of students who are starting from the beginning, students who are doing their PhD research and would want to do something that would help them publish, or for faculties who are working on some research projects that they want to publish or they want to apply for more funding. So we have been working with these researchers, and we have been helping them through our Omics Logic research programs. So some of the examples, like during the last program, that resulted in numerous independent research project proposals. So the project proposals were then peer reviewed, as I was telling you, and some of them include the analysis of microbial ecology of the International Space Station, and then implications of long-term human missions on Moon and Mars. So thinking about how metagenomics analysis and how these samples that we're collecting, giving us information to get a better living or to improve the challenges that we are facing by understanding the environment around, by understanding the microbiota. So I'm really excited to be a part of this collaborative research program and meeting you all here today. And I'm looking forward to meet the participants who decides to join the program and to talk more about the program and the registration part. Let me invite Deepsha to guide you through that process. But I'm here. I'm available to you, and you can always reach out to us for any kind of questions regarding research or projects that you have interest in this area, in this field of metagenomics. Thank you.
Thank you so much, Dr. Mohit. Now, I would like to draw your attention to the program resources. For different parts of the session, we will leverage Omics logic resources that you can use to learn about metagenomics. These will include asynchronous coursework with theoretical material, quizzes, practical assignments, our cloud-based analytical server where you can analyze large metagenomic data and visualize results, the Omics Logic Code Playground where you can practice coding in R and Python, as well as a repository of project examples. We will start with introductory coursework in metagenomics, where we will familiarize ourselves with the world of the microbiome. The modules will also explain key terminology and some of the project datasets we will use in the analysis. Some of the practical analysis approaches we will cover in this program includes kind2, data to pipelines, and phyloseq. Throughout the course, you will be given directions to access the bioinfo platform for bioinformatics processing and analysis of metagenomic data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. Different stages of analysis are performed in this different section of this multi-omics platform. The visual interface of the tBioInfo platform will enable users to quickly build logical graphs like you're seeing on the screen of their analysis and understand each step significant in running the pipeline. For those interested to implement the pipeline on their own, we will also offer guidance and references that can be used to effectively install all the required packages for the pipeline. Behind many of the scripts written in popular coding languages such as R and Python, these are also explained in tutorials you will have access to on our Code Omics Logic platform. These include beginner tutorials on data importing, preparation for analysis, and standard visualization of processed data. More specialized packages with publication-ready outputs will be covered in more advanced lessons. That is why this program is built to provide students of all backgrounds with a clear perspective on big data analytics, machine learning, and artificial intelligence. Our Omics Logic step-by-step process is enabled by access to complex tools via a simple and intuitive analysis platform, at the same time exposing participants to analysis logic in the context of real data sets, as you have seen Dr. Mohit explain, and extrapolation of this logic to a variety of projects and data sets with assistance from mentor and technical support. The first step, therefore, is to register yourself and start updating your profile like you see the participants doing here. Here are also a few examples from previous participants who have taken part in the Omics Logic programs, reviewed these programs, and also proposed novel research projects. We are excited that this event has been led and organized by a dedicated team in Nigeria that brings together experts who have an interest in data-driven research and have all recently completed our specialized training. They are now certified and ready to assist all of you in joining the program, getting registered, finding all the necessary resources, and helping you troubleshoot any technical or conceptual challenges you come across. We have Adekunle Adelowoye, who is our Omics Logic project lead and a faculty at Leeds City University in Nigeria. Adedayo Aburisade is the Omics Logic Community Manager for Africa, and he is a faculty at the Department of Oral Diagnostic Sciences at Bayero University. Sani Hamza is a medical laboratory scientist at the University of Lagos in Nigeria. I would now like to invite Adekunle to say a few words about the programs that we are running in Nigeria and his experience. Adekunle, over to you.
Thank you very much, Lisa. I'm very excited to be here this evening to be speaking with us. Adekunle Adelowoye, like Lisa introduced, I'm a faculty with Leeds City University here in Nigeria, and I'm also a mentor and program director with the University of Nigeria. I want to appreciate Pine Biotech, for programs such as these Omics Logic Metagenomics training, which is being availed participants across the globe, particularly here in Africa and Nigeria, in order to gather relevant bioinformatics skill sets that is relevant to metagenomics, with particular focus on hands-on experience, that would help participants, to benefit a lot and be able to apply this to their own personal project. I'm glad to say that the feedback we've had, from previous programs we've been running, including some which are currently ongoing, has always been an exciting one. It gives us in the team here in Nigeria a lot of joy to see that participants get value for the money they pay for such programs as this. And one of our goals, of course, is to see participants go from little or no skills as at the time of beginning trainings like this, to being able to learn relevant skills and applying same skills, to their own personal project. Okay, so with that conviction, of course, I have no doubt that for as many participants who will be joining this present Omics Logic Metagenomics training, we equally have a lot to benefit. Okay. We at Omics Logic Nigeria, we are very excited to provide all the needed support and mentorship, as well as support that is necessary for participants to get the very best, from these forthcoming metagenomics program. And so I would want to pass it on to my, our community manager here in Nigeria, and Dr. Adedayo, to equally have one or two, to say one or two things. Thank you. Dr. Adedayo, over to you.
Hello, good evening, everybody. Thank you for coming here today. My name is Adedayo Aburisade. I'm a consultant oral pathologist at the Kano Teaching Hospital in Kano here. I'm also a lecturer at Bayero University, Kano. And I'm the community manager for Omics Logics Nigeria, and also for Pine Biotech USA. We are very glad to have you with us, and we're also very glad to partner this with Dr. Elijah. This new metagenomics course, we promise that all your questions, queries shall be duly answered, and whatever queries or questions you have, you can duly send it to africanpile.bao. You get prompt response, prompt registration, and prompt answers to all your queries. We thank you and we wish you join us to make this community more formidable and to be avant-garde in metagenomics in Nigeria, in Africa as a whole. Thank you very much. I'll pass it over now to Bipsa. Hello, Bipsa.
Thank you so much, Adikunle and Aditayo, for sharing. I would now like to invite everyone to get started by registering yourself on EduTBioInfo. This is the first platform that we will all utilize for the program. So there is a free registration. If you click on the register button, once you are edu.tbioinfo, Aditayo, if you can kindly share the URL in the chat box. And if you're already logged in, I will be showing you the next steps on how you can register. So please go with me to edu.tbioinfo. And if you have not yet registered for the program or joining us for the first time, you will have the register button right here. And there will be a pop-up where you can enter your email ID and set your password. And you can log in to the website. Thank you so much, Aditayo, for sharing. So let us go and see how this program is going to be conducted. So this is what we call the organization page for the Metagenomics program in collaboration with Helix Biogen Institute. In this page, I am posting the link to this page. So if you're already logged in, you'll be able to have an overview of the page, though you will not be able to access the full page. Here you will find the events, the upcoming events of the program. So as Dr. Mohit has already explained to you, we are starting on the 16th of February. So you have about 2 weeks to join this program. Now, talking about the registration process, this is the direct link to register for the program. If you're already logged in to the portal, you will be able to access this direct link. And you can see here the details of the program. Now, the cost of the program is $150, but we are giving a special discount for our participants who are joining us. And to avail that 50% discount, we will be sending everyone an email with the code, and that will make the $150 to $100, which will give you a 50% discount. And in case there are students who are not able to afford the $100 program, we also have a scholarship option, which we will again share via email. And let me explain the scholarship option to you, the way the scholarship works. Whenever we have the scholarship option, we ask the students to go ahead and meet with us along with one of their references. So if you're applying for the scholarship program, you will have to mention the name of 3 of your references and to reach out to them so that they can join us for a meeting. Based on that meeting, where we can talk to your reference as well as you and learn more about where you're coming from and how much time you can dedicate to this program, you would be offered that scholarship. So if you are wanting to join this program and have an interest in metagenomic research, then please start applying for the program as soon as you receive that email from us. And moving on, I would now like to open the chat for any questions that you may have and invite you all to join the program. And as I did note in the chat, there are a lot of participants today who are also interested in infectious disease research. So I would also like to draw your attention to another program that we are all having on bioinformatics for infectious disease research as part of, in collaboration with Noguchi Memorial Research Institute in Ghana. So if you are a researcher or a student interested in infectious diseases, you can also check out this program, which we are starting from tomorrow. And I'm posting the link again in the chat box. You will receive all of these details, and all of you who have shared all of these details with us, we will reach out to you personally via email. So please make sure that you have also added your email IDs in the chat box so that we can reach out to you personally. And now I want to open this for any questions that you may have. And as you are asking all of these questions, I would also want to invite you all to go ahead and join our social media groups.
Okay, thank you for sharing your details here. I wanted to mention that one of the reasons why BIPSA asked all of you to complete the free registration on the educational portal is so that you can gain already access to the free course called Introduction to Metagenomics. This free course is going to introduce you to the basic concepts, terminology, and applications, just like what we did today in this session, but in a more expanded way. And we hope that by joining that course, you will be able to have access to our additional free resources that might be relevant to you. So I would like to make sure that all of you have tried that. And if you come across any issues, you're welcome to email us at BIPSA or marketing@pine.bio or Africa@pine.bio, specifically if you are in Nigeria or in Africa in general. And also, we mentioned the social media groups. And the reason why we ask you to join the social media groups is because we see this program that we're going to be doing with Helix Biogen Institute as one of the steps in building a community of those of you that are interested in bioinformatics in application to biomedical and agricultural biotechnology challenges. So we hope to stay in touch with you long term and really get to know all of you, as well as the institutions that are represented here, and develop a sense of what are the most practical solutions and training that is needed for all of you that are participating in these free webinars. Thank you.
Thank you, Elia. And thank you everyone for joining us today. We look forward to having you with us in the upcoming session. Please reach out to our team at marketing@pine.bio and Africa@pine.bio for any questions related to this upcoming program. And all of you who have shared your email IDs in the chat box, we will be reaching out to you. And if you have mentioned any specific interests, we will also be reaching out to you regarding all the resources that we have is per to that specific interest. So thank you again today. If you do not have any more questions, we would like to end today's session.
Okay, great. I see actually there are some people here sharing their details from Noguchi University and University of Ghana. So it's great to see those of you. You probably received emails, or you will receive emails about our upcoming program there. Yes, and all of you in Nigeria, please make sure to connect with our team on the ground in Nigeria with Africa@pine.bio. They will be following up with you with some additional details about this upcoming program. Thank you, BIPSA. Yes, thank you everyone for joining. We look forward to seeing you in the program itself, and we'll be emailing you the recorded version of this webinar. Okay.
Just to add one more clarification, the discount for the program is $50, which is a 33% discount. I'm sorry if I mentioned that as a 50% discount. I just wanted to put it out there. So we will be sending you the discount codes and everything via email that you will be receiving after the webinar. And also anybody who has mentioned a specific interest in infectious disease will also send you the details